Hello, my name is Wen Wang, and I am a researcher at the University of Minnesota Twin Cities. My research mostly focuses on developing computational methods to explore how genes interact with each other to increase or decrease our disease risk. First, thanks for stopping by my poster. Please leave a message if you have any questions. Today, I'm going to introduce my recent work on discovery of common genetic interactions across neurodegenerative diseases. Parkinson's disease and Alzheimer's disease share many similarities. They have similar cognitive and physical symptoms. They also share mechanisms such as alpha-synuclein protein nicotinic receptors. GWAS has been productive in finding loci that are associated with PD and load. However, the discovered loci only can explain a small portion of the estimated genetic contribution to disease. At the same time, existing studies have found evidence for genetic interactions in PD and load, such as Park16 interacts with LOCK2 in Parkinson's disease. IL-6 interacts with IL-10 in Alzheimer's disease. But due to a lack of statistical power, most GWAS studies focus on searching for single variant associations. So in this study, we'll use 6 PD and 5 load GWAS cohorts to study genetic interaction. In complex disease, genes often interact with each other to influence disease risk, which means loss function of individual genes might not have an impact on disease risk. But when a second or third gene also has a loss of function, then there can be an increased or decreased disease risk. In GWAS, because there are millions of genetic variants, which makes discovery of pairwise SNP-SNP interaction is quite challenging. Our lab has been working with Boone's lab at the University of Toronto to study genetic interaction in yeast for more than 15 years. What we learned is that genes often interact with genes with similar function. From the yeast genetic interaction network, we observed densely connected genes belong to the same functional module or between two functional modules with some coherent functions. Motivated by this observation, we developed a method called bridge to search for genetic interaction based on functional modules such as pathways or protein complexes. The input of the bridge method are GWAS data and a set of pathways. Then we compute pairwise genetic interactions based on cases and controls and construct a SNP-SNP interaction network. From the interaction network, we can evaluate if the within pathway model and the between pathway model and the between pathway models can be enriched by SNP level genetic interactions. Then we shuffle SNP pathway membership and the disease phenotype labels to evaluate false discovery rates. The output of the bridge method is a set of pathway interactions for giving FDR cutoff. We applied bridge to the 6 PD and 5 load cohorts. We first report discoveries that have FDR less than 0.25 and can be replicated in at least 2 independent cohorts. For PD, we find 20 WPMs and 47 BPMs. For load, we find 13 WPMs and 32 BPMs. Then for these discoveries, we further evaluate if they can be replicated in another disease. Using the same criteria, we find 9 WPMs in common for PD and load, but none for BPMs. So we relax the criteria, only require replication in 1 independent cohort and get 15 BPMs. Clearly, WPMs have better signals in this dataset. In this table, we listed example discoveries that are specific to each disease. For PD, one interesting discovery is keratin sulfate degradation. Keratin sulfate has roles in neural development and repair. The two top driver SNPs can be mapped to two genes, GLNS and ACAN. The ACAN gene provides instruction for making a protein called aggrecan. This aggrecan protein plays an important role in regulating adult brain plasticity. For load, we find a pathway called interferon gamma signaling. Interferon gamma has evolved in Alzheimer's disease progression, which is also interesting. So, other pathways on this list also look promising and, which is very encouraging. Let's look at the common pathway-level genetic interactions supported in both PD and LOD. I'm going to pick 3 within pathway interactions to discuss. First is the natural killer cell-mediated cytotoxicity. Natural killer cell has been widely studied in both PD and LOD due to its key function in the innate immune system. Studies have showed that NK cells has pathological role in both PD and LOD. We also found HRF1 alpha transcription factor, which is identified as a promising therapeutic target for treating neurodegenerative disorders. Emerging evidence also suggests beta cells also contribute to pathogenesis of neurodegenerative diseases. From the above discussion, you can tell many of our discovered pathway interactions are quite relevant to the disease we studied. More importantly, most of the pathways we discover cannot be discovered by previous single variant-based approaches, but they can be connected to previous non-GWAS experimental studies, which is very exciting. Next, I'm going to use an example to show we can apply some downstream analysis to gain more insight from our discoveries. This again is a within pathway interaction. The pathway is ADORA2B mediated anti-inflammatory cytokines production. First, this interaction is statistically significant and reproducible in both PD and LOD. We can identify driver SNPs and genes that contribute to the discovered genetic interaction. We list the top 5 SNPs and the genes mapped to them. We found that even the top SNPs are different, but the genes mapped to them are quite similar. So we plot the gene level interactions that are supported in both PD and LOD. In the end, we also can derive a polygenic score based on pathway level genetic interactions for each individual and use it to predict disease phenotype. So this 2-box plot shows that the polygenic score can differentiate disease status in both PD and LD. So potentially we can use this to make a prediction model for the disease. In summary, our method can find statistically significant and reproducible pathway level interactions and gene level interactions for both PD and LOD. We found evidence for common genetic interactions underlying both PD and LOD. Our preliminary results on polygenic scores also suggest incorporating interactions in individual risk prediction models can be very important. Okay, this is the end of my presentation. If you have any questions or comments, please feel free to contact me. Thanks for your time and have a wonderful day.